All right, really happy to be here. As Dana said, my lab has worked on development of the heart and the genes of muscle development for years, but with the advent of CRISPR, about 4 years ago, we decided to ask whether we could really do something about genetic disorders of muscle and apply CRISPR to an area that we knew a lot about. So to put this in context, it's really humbling to realize how many monogenic disorders there are that affect muscle in hundreds of different genes, and there's not a single cure for any one of these. This is a major area of unmet medical need. The Mount Everest of muscle diseases, the biggest one of all, is Duchenne. Duchenne affects 1 in 5,000 boys, so there's 300,000 boys in the world right now afflicted with this disorder. It's the most common fatal genetic disorder diagnosed in childhood, and there is no cure. So the way in which this disease is typically diagnosed, I receive emails every single day now from mothers around the world, and they usually tell me the same story. So they notice that their son is having difficulty walking or keeping up with his playmates on the playground. They take the son to the pediatrician, they do a series of tests, and if the ultimate diagnosis is Duchenne, DMD, there is no escape of the clinical progression of this disease. So no person has ever escaped the consequences of a mutation that leads to this disease. So these boys are wheelchair bound by about the age of 10. They lose ambulation and have limited use of arms. They ultimately become confined to a ventilator because breathing requires your diaphragm, which is a muscle. And although not widely appreciated, the ultimate cause of death in these boys, which occurs typically in the mid-20s, is heart failure due to disintegration of the muscles of the heart. The disease is caused by mutations in the largest gene in the human genome, the dystrophin gene. This gene was cloned by Lou Kunkel more than 30 years ago, and despite intense efforts over decades, there has not been a cure for this disorder, as we'll talk about. The mutations that cause Duchenne are transmitted on the X chromosome. That's why the disease is largely confined to boys. This gene is massive. It encompasses 3.5 megabases, so for the people interested in gene expression, put that in context, it takes about 18 hours for RNA polymerase to traverse this gene one time. So it basically takes you all day to make one copy of the RNA, and it has 79 exons. So here's one of the challenges. There's more than 3,000 mutations that have been identified in the dystrophin gene, and it raises the question of how can you conceive of a gene editing therapy with so many different mutations. And I'll tell you about an idea that we had to consolidate many of these mutations. So this shows the structure of the protein encoded by the dystrophin gene. It functions as a molecular shock absorber, schematized here, for all the muscles in your body, every muscle fiber, including the heart. And it does so by connecting a cell adhesion complex at the membrane called the dystroglycan complex with the underlying actin cytoskeleton. And you can imagine that this is required to stabilize the structure of muscles during the continuous contraction of the heart and of all the muscle fibers in the body. Now, I should say that the disease is typically inherited from unknowing mothers who are carriers. They don't realize they're carriers of these mutations until they have a child with the disorder. But that accounts for only 2/3 of the boys with Duchenne. 1/3 of boys with Duchenne acquire the mutation spontaneously. And for that reason, this disease can't be eliminated by germline editing or pre-implantation diagnosis. This disease will always exist for that reason. This is what the dystrophin protein looks like. I'll show you several slides like this. This is an immunostaining of a muscle group, and you can see every muscle fiber is underpinned by this molecular scaffold. Now, here's the schematic diagram of the protein at the top. The two ends of the protein are essential, and this is an important point. One end anchors it to the cytoskeleton, the other end anchors it to the cell adhesion complex. The intermediate region of the protein is comprised of a series of rod domains, and these are like coils of a spring that provide the elasticity. And we know that all these rod domains are not essential. In fact, you only need 4 of them to maintain residual function. And we know that because there are patients in the world who have a form of muscular dystrophy called Becker muscular dystrophy. Some of you may have heard of that. It's a relatively mild form of the disease. And these patients have in-frame deletion, so they have the 2 ends intact, and they have a subset of the coils, but they're missing some of the internal regions. And these patients can be fully ambulatory into their 60s or 70s, and so that tells you that you don't really need the whole part of the protein, the whole internal part of the protein. Now here's the structure of the exons, and the shapes of the exons. If they fit together like pieces of a jigsaw puzzle, that indicates that splicing maintains the open reading frame. This is obviously a really complicated gene, so nature did not fool around with it once it was invented, and the structure of the exons and even the splicing patterns are conserved across vast phylogenetic distances. This is good news. You can do a study in a mouse and you can extrapolate directly how it would work in a human. So although there's more than 3,000 mutations, they cluster into hotspots, and these 10 exons in red are the major hotspots, and they account for almost 80% of the mutations in this gene. So if you could devise a genetic strategy to skip over mutations in this region, we reasoned you might be able to apply this to large cohorts of patients, and that's what I'll talk to you about today. Now, over the past 30 years, as I said, there have been many, many attempts to correct Duchenne, and this slide summarizes basically everything that's been thrown at this disease, but to no avail. There's been one drug that's been approved for treatment of boys with Duchenne. It's an oligonucleotide that can induce exon skipping to bypass one mutant exon, exon 50, which I'll talk about more. It was approved by the FDA about 2 years ago based on a clinical trial in which that oligonucleotide was infused through an indwelling catheter into boys for 1 year. And at the end of the year, they were required to measure the amount of dystrophin protein that could be restored by exon skipping, and the amount that could be restored was less than 1% of the normal level with an error bar of greater than 1%. So the good news is that you don't need to restore dystrophin protein to 100% to cure this disease. It's estimated that if you could generate 15% of the normal level of dystrophin, that you could cure this disease, but 1% is probably not enough. That drug, though, is currently on the market. It costs about $1 million per year per patient. So the cost basically the cost to make dystrophin in those boys is about $1 million a year for 1% dystrophin. So, and it hasn't been efficacious yet. So we decided to come at this disease from a completely different approach. Let's not treat the symptoms, let's not treat the consequences, let's treat the cause, the molecular lesion that leads to the disease, and we know what that is. We know what all these mutations are, so we decided to try to ask, could we apply CRISPR to correct Duchenne? And we thought about this for a long time. We tested a lot of approaches, and we ultimately settled on what we called single-cut CRISPR gene editing, and that's schematized here. So here's a portion of the gene, exons 49 through 53. The most common deletion in boys is the deletion of exon 50. These are the same boys for which the drug I just described to you is applied to. And we reasoned if we could design a single guide RNA that could cut in exon 51, perhaps it could lead to either of 2 consequences. If it made an indel that deleted the splice acceptor of exon 51, the splicing machinery is agnostic. It just looks for the next splice acceptor, and so we saw that 49 could go to the next exon. 52, the shapes are compatible. It could put it back together by exon skipping. Then we thought maybe we could make a really small indel in exon 51 and reframe the trinucleotide reading frame with the preceding one, and that would be the most minimal possible correction. So here is a big reason that we got into this. This is my neighbor. His name's Ben. He went to school with my kids in Dallas, and you can see he's in a wheelchair. So Ben was the first patient that provided us with a blood sample to enable us to test our ideas. So here's what we set up. We set up a Duchenne clinic program sponsored by the NIH in Dallas, and patients can come in, such as Ben. We take a small blood sample, 4 milliliters of blood, and we can convert them to iPS cells, and then can convert them to cardiomyocytes, and this is what they look like. So for those of you who haven't seen these before, these are cardiomyocytes. Those are actually Ben's cardiomyocytes that we made from a sample of his blood, and they beat spontaneously. And then you can treat with a guide RNA directed towards the mutant exon in these patients. And it's a very powerful approach because you can test whether this guide RNA in the genome of the person with the mutation that you're interested in can restore dystrophin. And if it can, is that dystrophin functional? Does it go to the right place? Is it stable? And then we have a number of functional assays we can use. So here's what happens. These are cardiomyocytes from a Duchenne boy stained in red for dystrophin. They don't make any dystrophin. We made a single guide to correct the mutation by exon skipping, and they're converted to dystrophin positivity. And here's a Western blot, protein blot. So here's a normal boy. This is the Duchenne boy. This is actually Ben's samples here before we edited them. It doesn't have any dystrophin, and then we correct it, and dystrophin comes back. Now, the power of this approach is that when you make the correction, you're making it in the genetic milieu of the endogenous gene, so the corrected gene is then expressed at the right time, the right place, the right level of the endogenous gene. This distinguishes this approach from gene replacement therapy, where you add back a gene in a virus and you're dependent on the longevity of expression of that virus as well as whatever cis-regulatory elements you put into that virus. In this case, once the gene's corrected, we believe you're good to go. So here's the structure of the gene. This is the most common deletion, exon 50. 13% of the boys in the world have that particular mutation. So we wanted to see if we could correct this, not just in cells, but in an animal model. There wasn't an animal model, so we made one by CRISPR. We deleted exon 50 from mice. And so here's what those mice would look like. They have no exon 50, so they can't make dystrophin because it's out of frame. Then we had to figure out how can we deliver the gene editing components systemically. You can't do this by cellular replacement because skeletal muscle is 40% of your body mass, and one thing we've learned in the cardiology field in the last decade is stem cells don't colonize the heart, so you can't put cells back in the heart. You have to fix it in situ. So we turned to a virus, adeno-associated virus, AAV9. I think you've all heard of it. This is a harmless virus, and it has tropism for muscle tissue. And we engineered into the virus, after a lot of trial and error, we engineered 2 viruses. One virus encodes Cas9, controlled by a muscle-specific promoter called the CK promoter. And this restricts the expression only to muscle. So even if the virus inevitably will go to the liver, but it won't express there because the Cas9 won't express there because this is completely muscle specific. And then we made a second virus that has a single guide. We call it single guide 51 because it's directed against exon 51. And we use 3 different Pol3 promoters to direct its expression. I can talk more about that later, but remarkably, as we got into this, we figured out, I always thought Cas9 would be limiting for editing, but it's not the case. The limitation for efficiency of gene editing is the guide RNA, and so the guide RNA has to be in excess over Cas9. So here's what we found initially in mice. So the top shows 3 representative muscles. Triceps is the muscle of your arm. Here's the diaphragm. Here's the heart. All of them express dystrophin. Here's the mouse model with the human mutation. No dystrophin at all, so it's really a reliable model for the pathogenesis of the disease. And there's what happens if you inject these mice with CRISPR-Cas in AAV and you look 8 weeks after 1 dose, it restores dystrophin through every muscle that we've looked at in these animals. Dystrophin expression is restored. Now, an important question was, what is the molecular basis for this restoration, and what is the actual CRISPR edit that's occurring? And so we, as I mentioned earlier, we felt we could do it by skipping. We wondered maybe there could be reframing, we really needed to look at that, and here's where we got a big surprise. So we did RT-PCR to look at the transcripts for dystrophin in the muscles from these corrected mice, and 15% of the transcripts were not corrected. Not surprising, it's not 100% efficient. So in those uncorrected ones, 49 goes to 51. They're out of frame, so you can't make dystrophin. But 60%, the majority of transcripts in the muscles of these corrected mice, all had the same correction, and it was always a single nucleotide insertion, an adenosine insertion, immediately downstream from the PAM sequence. And fortuitously, if you put a single A insertion at that site, it reframes the exon and puts it in frame with exon 49, so now you have the contiguous protein. It's the simplest possible molecular edit you could make to restore dystrophin. And when we first saw this, we didn't believe it. I absolutely didn't believe it. And then we went back and looked in human cells with the same guide, and we saw exactly the same insertion. And then, as you'll see in a minute, when we did it in dogs, we saw the same insertion. So this particular guide has a propensity to lead to that type of correction. And then 20% were skipped. So these had a larger indel that deleted the splice acceptor site, that's schematized here, and so you got skipping from 49 to 52 and still gave back the dystrophin protein. The single A insertion is interesting, and when we first found it, nobody had reported that kind of a correction, and subsequently several groups reported that there can be single nucleotide insertions at Cas9 cut sites. And we think it occurs like this. So here's the PAM sequence, Cas9, when it cuts at that particular site with that particular guide, it leaves a single nucleotide overhang, and the DNA repair machinery fills it with an adenosine single A insertion, and that reframes the entire protein. So we've been able to restore dystrophin very efficiently in these mouse models of the disease, and that not only restores the protein, it restores the muscle function and throughout the life of those mice. So we wanted to ask, is it possible to ramp this up into a larger animal? We searched the world over, and we found one, and there's a colony of dogs, beagles, at the Royal Veterinary College in London, and they have the same mutation as the most common mutation in boys, the exon 50 deletion. Makes sense? I told you the gene is really conserved, so the fragile regions of the gene where mutations tend to occur are also conserved. So this colony is maintained by Professor Richard Pierce at the Royal Veterinary College. We scaled up and put CRISPR-Cas into AAV and went there to treat these dogs systemically. No one had ever treated a large animal like this body-wide with CRISPR, and frankly, I was kind of very worried. I didn't know what was going to happen. But so we went and treated the dogs, and they didn't skip a beat. Everything seemed fine. So here's biopsies of muscles from normal and from the dogs with the same exon 50 deletion. Here's what we saw after 8 weeks. We could restore dystrophin. Diaphragm's a really important muscle to restore it in biceps and heart. I told you that quantification is really important when looking for drugs or gene therapies that are going to replace dystrophin. So we quantified the amount of dystrophin protein that could be restored in these dogs, in these biopsies, and here you can see that. So in the left of each of these protein blots is the normal dog. Here's the dog with the disease, and here's the corrected dog after 8 weeks of one dose. So 92% of the level of dystrophin protein is restored in the heart. Here you can see the diaphragm, and here you can see the biceps. So this method can really work, and it can lead to sustained expression of dystrophin in an animal that has never expressed it before. And this was something that I was really surprised about. What if you take an animal, they've never made dystrophin, and now you put it back, can it do what it's supposed to do? And it goes to the right place, and it seems to do the right thing. We started with a single patient sample, Ben, my neighbor, and we've now extended this to all sorts of mutations. So boys come from all over. they write to us every day. If they have a mutation we haven't corrected and they're local, we will obtain a blood sample and we'll test whether we can correct it. If they're in a faraway place, which is pretty common lately, and they have an interesting mutation, we can just recreate it in iPS cells and let them know if we can correct it. We've extended this now. We've made mouse models that have mutations that account for 80% of the Duchenne mutations in the world, and we've corrected the majority of those, and we're still working on optimizing the guides for those, and as you've seen, we've scaled it up to dogs. So looking ahead, these are the issues that we're currently confronting. Off-target effects. So we've looked for off-target effects by deep sequencing of predicted targets and are now doing other approaches. So far, we've not seen off-target effects above background in these animals, and my own feeling, my hope, is that there probably aren't going to be major off-target issues with this approach because muscle is permanently post-mitotic. Muscle cells don't divide. The nuclei and genomes are really stable. And so issues like tumorigenicity or things like that don't exist. There aren't any such thing as muscle tumors. Immune response, we're looking at that. We've not seen an immune response so far, although this is certainly something we need to think about because Cas9 is foreign protein. All the boys with Duchenne are on immune suppression with corticosteroids, so that may be mitigated. How long does it last? Will it last forever? Muscle pretty much your heart lasts forever. The heart cells you're born with are the ones you die with, and skeletal muscle is really long-lived. We know that in mice it'll last the lifespan of a mouse, but a mouse only lives 2 years. So I don't know if it'll last forever. We need to look at that. And we're currently testing new enzymes, delivery systems, and trying to extend this to other diseases of muscle, taking the lessons we've learned from this approach. So to transition this from my laboratory to address those sorts of practical issues, we initially started a biotech company called Exonics, and it was recently acquired by Vertex, and they're now applying their knowledge of gene editing and gene therapies to treat Duchenne. So we have been embraced by the patient community. These are all boys with Duchenne. These boys here are from Austin, Texas. They're brothers. They both have Duchenne. That's one of the issues of this disorder. So boys are typically not diagnosed until they're 3 or 4. It's not uncommon for a couple to have had a second son before the first one's diagnosed. So there are many families with 2 boys that have Duchenne. And we've corrected many of the mutations in these boys. So the patient advocacy world is huge for a disease like Duchenne. They really have embraced our vision. This is a recent patient advocacy group where I spoke. And all those boys have Duchenne, and that's their families. So it's just a wonderful group to be around. And these are the people that did the work. So our work was started by Chengzu Long, a student in my lab, and Leonela Amawasi and Yili Min were a postdoc and student in my lab. They're now working at Vertex to take this work forward. We've had great collaborators as well as support from the National Institutes of Health. And with that, I'll thank you for your attention.